Okay, so we're going to start by going through the syllabus and talk about what the format of the course is and everything. And then, as I said, I will answer your question. You must have all gotten an email from me with a link to the syllabus. It's in a Google Doc because we expect to make a few other changes to it, and we still don't have all the details right. But we'll go through it. And don't print this out. Just basically look at the Google Doc if you need to know what's going on. So as I said, the course is the cognitive neuroscience. We meet on Tuesdays. We start at 6 o'clock. We go until about 7:50. We're going to take a break in the middle. This is the room that you all founded. I think it's a much better room than the one we had previously. So that's a good thing. I am Hussein Adeli. I'm the instructor for the course. I am part of the new school as a part-time faculty, if you know what that means. It's basically a teaching faculty. My main affiliation is with the Zuckerman Institute at Columbia. I'm putting my office hours to be right before the class. At this point, it says the lounge, but it's likely to change. So as I said, just look at the syllabus. We have 3 TAs for the course. Olivia and Ellen are here. We'll get to meet them as the semester goes forward. We also have Nadia, but she can't be in the class, but all of them have TA'd for the course and have taken the course previously. So they're great resources and they're the nicest people on the planet. So you should definitely reach out to them if you have any questions. We're gonna update their office hours and stuff. I've asked them to have maybe an hour of online office hours, an hour of in-person office hours, so you could all reach them. We'll divide the class among the 3 of them. So you will have a dedicated TA and all your questions and concerns should go to them. You should not email 4 people with a simple question. If you have anything, you have to email your TA. What we've done previously is that I just let you choose who you want to work with based on whose office hours matches yours better in terms of availability. We'll probably do something like that later this week. Any questions so far? Okay, so you all know what this course is about. You've read the description before taking the course. We will go through some basic neuroanatomy and physiology of the brain. We'll also talk about function. We'll also talk about specializations of different brain areas. We'll talk about behavior, actions. It's an introductory course, but it's also a graduate course. So there's depth and there is breadth. We'll cover a lot of this stuff and we'll try to get deeper on some of them. Hopefully by the end of this course, you'll be able to pick up any paper about brain sciences and read it and understand it, or know how to find resources in order to understand it. That's the objective here. There's a textbook, and I call it optional because everything that will be on the tests will be covered in the class. If something's in the book, but I didn't talk about it, it's not going to be on the exams. That's what optional means. On the other hand, if you're someone who wants to get deeper on a certain material, or you don't have the right background for this course, you should read the book. It helps you understand the material much better. I put a PDF copy of the book on Canvas. You all have access to it. You don't have to buy it. So use that. The grade in this course basically is divided in three categories. We have exams, we have assignments, and we have a final paper. There are 2 assignments. One of them is writing. You will write your reaction to a paper. The other one is you basically presenting a paper. Each of them is 10%. we'll have all the instruction here for each of the assignments, how you would go about preparing it. Um, you should look into it more. Again, these are all available on the syllabus. The final paper is also something that you'll write as we get closer to the end of the semester. You'll pick a topic, you'll check your topic with your TA, and then they'll guide you through that process. And then when you have a topic, you could start working on it. So we have a late submission policy. Sometimes we don't impose it, but I think we should do it this semester. We want you to be on time with things. If you have to submit something at a certain time, do it. Um, Obviously, we'll understand and we're open to give you more time with different things, but you should always try to be on time. I think for the assignments, it's something like um, if it's not in the system by the time that it has to be in the system, you lose 10 points, and then for every day that you're late, you lose another 2 points, and all the grades are from 100. For the final paper, there is just a hard deadline because we have to grade them and put that. So we cannot give you more time unless you want to get an incomplete or something. So you should always try to be on time, to be early, and start your work earlier. Any questions? So the exams are 60% of your grade. We'll have 3 of them throughout the semester, and they have both short essay questions and multiple choice questions. The short essay questions will come from a study guide. We'll give you a set of questions and we'll pick some of them for the short essay. However, there are also multiple choice questions that could come from anywhere in the material that was covered. It does not have to be part of the study guide, but it will be something that we covered in the class. So you should study the material. You should also study and work on your study guide. But you can't leave anything out. Extra credit, this is just something that, it's important, is that you be a good student, and you know what that means. We know what that means. It means that you show up, you participate, you're not late with your stuff, you're not, trying to bend the rules all the time, and also you're a good citizen. You're not, you're doing things the right way, and you're not complaining when, there's no room for complaining. The rubric is standard new school rubric. We have in total 103 points because of the 3 extra points. Anyone above 93 would get an A, and then the rest are here. You could check it out. This is something we used last year where I created a simple, very simple tool, if you could call it that, to put your grades in and then calculate your, so let's say these are your grades. You could just basically run, and I have the instruction in the syllabus. You could basically run this and then get your grade. Now, let's say you had an 80 here. You run it again, and you would get a grade like this. So this is for you to monitor your performance in the class. And based on the grade that you've gotten, based on the grade that you expect to get, you'll know where you stand. Okay? Pretty simple. Lecture recording, barring any technical difficulty, but today we'll figure it out. So I am recording this, and hopefully it is recording the right way. I'll post them. This is not to replace attendance. This is an in-person class. So you should always attend. I will make the recording available as a supplementary, as something that you could watch again if you have any questions, or you could study it as part of your exam prep. But it will not replace attendance. COVID policy will follow whatever the school wants us to follow. An important new topic that we have to discuss and be part of our syllabus is the use of generative AI tools that we're all kind of heard of. And again, the school policy is that the use of these things, the unauthorized use of these tools is prohibited. It's very clear. However, we understand that technology changes the way we do our work. And we will not want to completely prevent you from using them in this class of these. However, there are certain ways that you could use the system that make sense, that does not clash with the objectives of the course. And we want you to be in that vicinity. The reason for that, again, you hear that from a lot of people probably, is that we want you to practice writing. We want you to practice developing your voice, developing your way of thinking. And you don't want to just defer all of that to another system. So for that to happen, you have to do the difficult task of writing and developing your ideas and writing, and making sure that it's coherent and all those things. I have a few examples here of what I think could be potentially useful ways for all of you to use these tools. For example, you could, if you are not a native speaker, or if you are and you struggle with grammar, you could feed a paragraph to some of these method models and then ask them to fix it grammatically, right? That's a reasonable thing to do. You could maybe give an idea and ask for an outline. That's also a reasonable thing to do. You could ask it for a research topic. You could do other examples that I have here. However, if you do use this system, you have to have a paragraph at the end of your submission and explain very clearly how you used it, right? If you don't, we'll count that as a plagiarism. Also, you should know that these methods are very limited. They hallucinate, and they make a lot of factual errors, and they come up with random references and whatnot. You're responsible for all that. If you're making a submission with those errors, you're responsible for it. Under no circumstances, you should submit something that was entirely generated by a system as your own work. Any questions? All right. I assume if you're not asking questions, I assume these are all very clear. This is week 1. We're going through the syllabus. I'm going to talk a little about neuroanatomy and neurophysiology. Later this week, and again, I understand some of you do not have a science background or don't have a psychology or neuro background. To address that, Ellen will give a crash course type of thing on basic neuroanatomy and cell biology, Thursday or Friday.
I think Thursday afternoon, and I will record the lecture and make it available on campus. So if you can't attend, you'll have access to it. And I'll send out a reminder to the group tomorrow with the finalized schedule. So that'll all be available on campus.
Yeah, so again, it's going to be on Zoom. You should all attend it if it fits your schedule. Even if you have the background, it's a good refresher on the basic anatomy of the brain and body and how they interact. In this course, we're not expecting any hardcore neuroanatomy from you. We're not going to ask you on that or anything like it. But it's good to have the background. It's good to know what the basics are. Next week, we'll continue on neuroanatomy and neurophysiology. We talk about methodologies that are used in cognitive neuroscience. Each of these modules, which correspond to each week of the semester, will tell you what are the relevant chapters of the book that you could read to be prepared for this. It also may suggest you other reading material. Um, those are not mandatory. Those are for you to just understand the topic better. Third week, we'll again continue this topics. And then on the fourth week, which is September 19th, we'll have our first exam. And it will be, exams are not cumulative. So the exam will be on the material of the first 3 weeks, and then the following exams will not include this material. It will be the whole class. You're not going to need the whole class probably, but you'll have the whole class to take the exam. As I said, it will be a mix of multiple choice and essay questions, and we'll have them right here. Yeah.
For the exam, is it handwritten?
Yeah, handwritten. The exam will be handwritten, and you're not allowed to use any device or dictionary or anything. And we have tables here, so that's nice. Yeah. Other questions? Yeah.
How many questions in total roughly for each exam?
I'm thinking maybe we have 10 multiple choice and maybe 5 or so essay questions. Something like that. Okay. On that week, we'll have a couple of tutorials from Olivia and Nadia, the other two TAs, and they'll talk about how to read a scientific paper, because this is something we see with a lot of the students here, again, coming from non-science background or even from science background. They've never read a scientific paper. They've read books or they've seen, other ways of communicating science, but not necessarily sitting down with a scientific paper and reading it. So there will be a tutorial. There would be a couple of them on that. I was initially thinking of having this myself. I'll talk about it a little bit, just following on what they did the prior week. And the reason we're doing it then is because the next week you're going to have the first assignment. And the first assignment will be on basically reading a paper and then writing your reaction to that paper. You will be provided with a template. We'll give you a template. The template tells you basically a bunch of questions that you're going to have to answer, pretty much a paragraph to each of them. And the questions will be, well, describe what they postulate in your own words. What tools were used in the experiment? What were the salient findings? What were the limitations? What was the conclusions, right? So basically we want to see that you understood the paper. I'll give you a template for the way to organize it, but also I'll give you a couple of templates from last year to another paper that people wrote their reactions that was kind of nice in my opinion, and that would guide you through what level of details we're looking for. But as I said, you'll have a tutorial the week prior to that, maybe on Thursday or Friday before this week, on how to read papers. So you should start reading it a little earlier and then come with your questions to your TAs and be like, okay, I have a hard time understanding the method section. How should I go about it? However, this is a graduate course. We'll expect a lot of self-learning from you, right? We're not going to feed you everything. If it's a hard paper and you want to understand it, you have to research it. You have to go and then watch maybe a YouTube video on a certain concept. Maybe you have to do something else. You have to do a lot. It's not going to be telling you everything. You should be willing to do a lot of that research yourself. I'll add another paper or two here, so you have options to pick from. But for now, it's a little early for that. I'll do it later. We'll also review the exam. I'll do it. Previously, we did it, like the TAs did it outside of the class hours, but this year I'll do it in the class myself. Okay, so we'll have a few papers here. You'll pick one of them, and then you'll write your reaction paper on it, which is around like 4 pages. Like I think around 1,000 words would be nice. Maybe 3-4 pages, double the space. It's not really that long. Then we'll continue with our lectures. We'll the first module of the course is on neurophysiology and neuroanatomy and some basic um, in the terminology of neuroscience on low level stuff because uh, that's what they call it. And then we move from there to high level stuff, which is, for example, recognition, perception, memory, those things. So that's the trajectory we're taking, and it follows the trajectory of the book. All right. After another few weeks, um, we're also something we did previously, and I think it's a nice thing. You're gonna, when you submit your assignment one, you're gonna get a lot of feedback from your TAs. We'll give you the option to resubmit assignment one based on that feedback that you got. You could use it and then improve your submission, and then resubmit it a couple of weeks later. This would help you if, again, because you've never done something like that. This would um, help you do things the right, meaning that it gives you another opportunity, so you're not losing points just because you didn't know how to do it. So this is an option you have. And we'll pick the best grade of, you know, the resubmission will be better than your first submission. So that's the plan. We'll have our second exam on October 24th, and it will cover the material from the week 5 to 8 because it's, as I said, it's not cumulative. It will follow the same format as the first one. Then we'll have the second assignment. The second assignment is, again, you read a paper, but in this case, you make a slides. You present the paper. We don't have time to bring everyone up here. So the alternative is that you'll actually make these slides based on the paper that you chose. You present it. If you want, get a few friends to be your audience. But what we expect is that you present it, you record yourself, and then it would be like a 10-minute presentation, and then you upload it to a Google Drive or something, and then share that link with us, or put it on YouTube and share that link with us. We don't have enough space for you to actually submit a video. We just want a link to whatever you recorded. Questions?
Yeah, so you do it as a group project or
No,
is it?
individual. You could help each other out, but your submission would be individual. One way to do this is the way I'm recording this stuff here, which is through Zoom. You could just start a Zoom meeting, share your screen, then bring up your slide, put yourself in presenter mode, and then start recording. Then you start talking, and when you say start recording, you could record it to a cloud or your own computer, and then if it's recorded to the cloud, then you could download it. The problem with Zoom is that if you the cloud recording is going to expire after a month, so we don't want a Zoom cloud link. We want you to actually download it, put it on Google Drive or YouTube, which is stable, and then that's the link we want. But Zoom is a good way of recording your own. But since you're on Zoom, as I was saying, you could invite a few people to just watch you, so you could present to somebody. Then it would be the last module of the course. We have basically 3 of them. We talk about action, memory, and then we talk about some more high-level stuff like emotion, cognitive control, and if we have time, maybe consciousness. Then we have exam 3. Exam 3, so prior to this, I would put the exam on the last week, but it turns out there is I don't know if it's the same this year, but Max course comes right before this one, and you have exam there, and then I didn't want that to happen. So I'm putting the exam 3 the penultimate week, so you don't have to take 2 exams in one day. And also it gives us an opportunity to reconnect after the third exam, and you'll have the opportunity to see your exams, because every time we have an exam the following week, the TA will grade them and bring them to the class for you to see. So at least it gives us that opportunity to do that. We'll also do course evaluation in class on that day. So bring your laptop and then you'll do the evaluation of the course. We'll wrap it up, and then I'll answer any questions you'll have about the course, about the writing your paper. The paper has two parts to it. One is that I want you to submit your idea earlier. Now, your conversation with the TA should start prior to that. So maybe after your second exam, which is late October, that's when you should start, because you don't have any more assignments at that point. So you should start. You have an assignment here. So after November 7th or so, or November 14th, like early November, you should start thinking about your final paper, or even earlier. And you should start conversation with your TA, and they'll guide you through it. We'll have a little guideline thing that you made last year in order to help you narrow down your topic to something manageable. The format of this paper is like a review paper on any topic that you're interested in and relates to the class. And in a review paper, you basically want to learn more about a topic. If it's maybe the relationship on neurotransmitters and depression or some other pathology. So you basically go read 5 to 7 papers on that topic, and then you write a review of that topic. You basically start with an introduction that summarizes all those findings. Then you talk about different topics. What we don't want is that you just say paper one and then summarize it. Paper two, summarize it. That's not how to write a review paper. We have examples of it. And then when we get closer to that date, we'll talk about it more. But the idea is that you'll say, okay, how do these, so well, they could interact through this mechanism. So that's a section, another mechanism. Or maybe you could critique certain findings, be like, okay, limitations, right? You want to actually synthesize a paper, a review paper from those things that you've learned. You don't want to just go paper by paper. You'll submit your topic by November 20th. And this is before the Thanksgiving break. And then you'll have, the break to think about the topic, also study for exam 3. But yeah, any questions?
All the exams will be the same format.
Yeah. The exams all would be the same format. We'll have as I said, we'll have a mix of multiple choice and short essay questions. When is it due? The final paper is due December 8th. Now, it's because you have to have grades by a certain date. And I say we cannot accept anything after December 10th. If your paper is not in by December 10th, you're getting an incomplete. Because the TAs would have to grade them, and we have to have time to look through everything and then submit the final grade the following week. But as I said, not having the exam on the last week also helps, because you could take your exam on November 28th, and then you'll have the whole time until December 8th to work on your paper. But I hope that you're starting much earlier than that. It's a 10-page paper. All right, let me just go through this first. So there are some policies and whatnot, and you could look through them. They're boilerplate. The final paper, and I have an example here from a friend. Where the format is an APA style. And if you don't know what APA is, as I said, we have a tutorial in a few weeks on how to format your papers in an APA style. But it's simple formatting. You'll have a title up there, you'll have the page to start, and then you start with an abstract, and then you go through it. Now this one is where is memory information stored in the brain? Like that's a topic, right? This is a review. This is a topic that you want to learn about. Go study it. But you could pick something that is closer to what you care about and the stuff that is part of your bigger project or something like that, or open to you exploring those things. It would be 10 pages. I mean, this is a much longer one, but what we expect from you would be a 10-page thing, and then at the end you'll have some bibliography of the papers that you've cited and everything. Again, I said somewhere between 6 to 8 papers would be nice. We don't expect many more than you. Questions? I think we covered everything, right? All right, I have the information on the how to format it and stuff, but we also have a link down here on getting you started with the APA style in terms of general formatting and whatnot. So if you're not familiar with it, there are a lot of resources out there in terms of how to do it. Okay, and then you have the breakdown of the grade information. It's basically 20, 20, 60. And yeah, so one thing to note is that this is one thing about this class is the heterogeneity of people's background and prowesses and whatnot. So people come from different places, are going different places. So it makes it difficult to pitch the course at a certain level. There are, you know, this like anything in life, it's a bell curve, right? Most people are in the middle. There are people that come with a stronger background, or people come from weaker background. The course is catered to the middle. You know, it's to people who have some understanding of these topics and are interested in it. If you are at certain extremes, then the course may seem, I wouldn't say too easy, but it may come to you easier. And if you're on the other extreme, you may find the course very difficult. If you are there, try to use all the resources that you can in order to perform better. Like we are not assuming that you know a lot of this stuff, right? Everything you need to know will be covered. However, it requires you to really want to do well. It requires you to really want it. So I don't think there's a gap in here that you cannot fill with hard work. Any questions about the syllabus before we move on?
Are they allowed to use any of these as part of the 68 sources, or do they have to be
The
separate?
papers that we have in the class?
The final paper.
So any readings of what?
Like, so in some of the weeks we have like cited articles that are probably more.
Yeah,
stuff.
if it's not an assignment, then yeah, you could do that. If it's not something that you've done an assignment on, you could use it. Yeah. Just don't use a paper for assignment and for your final paper. Any other questions? Normally, we'll take a break in the middle, just because it's like a 2-hour long class, and I don't think we want to just go on like that. But let's just start a little bit, and then we'll take a break maybe in a little bit. I'll just say a few little things, and then. So with the lectures, my goal is to upload them a few days in advance, so you get a chance to look at them before coming to the class. So the one that we will go through is neurophysiology today. And again, if you struggle with some of these things, as I said, Ellen is going to give you a crash course. So don't panic. That's something that you should always remember. Don't panic with like if you think, oh, this is too difficult or something like that. There's a slow process and you'll get there in time. So neurophysiology, again, what does that mean? So a concept. So this is a simplification of what happens between an agent, an organism, and its environment. It's very simplified. But we're going to use that to understand certain concepts. So if you have an agent in an environment, right, you could, let's start from, down here, or from up here, from an environmental stimulus. You can see this right there. So there are a lot of things in our environment, right? There are sounds that we hear, the lights that comes, through our eyes, there's the things that we smell. And we'll talk about those different, what we call modalities, being vision or audition, or olfactory, taste, touch, right? These are different modalities, and you could perceive them based on your interaction with them, right? You're looking at me right now, for example. So that would be something like an attended stimulus, right? And a stimulus is just a fancy word for this sort of visual input, for example. Then the stimulus would fall on your receptors, right? The way that we perceive the world is because the light falls on our eye. If you close your eyes, if you block the light, you're not going to see the environment. If you turn off the light, the light here, you're not going to see anything in the environment because nothing comes to your eye. This energy that comes to your eye would be transduced. If there's a process of transduction, it becomes neural activity. This neural activity gets processed to become perception, right? That's how you perceive me as a human. Then you recognize me as someone that you may have seen before or earlier. Then you could act on the environment, right? You could move your head or you do something else, or you could look at me or look at something else. That's an action, right? And then you change what you perceive again, right? So this is a cycle. It's a simplified cycle because in many ways, that's not how we interact with the world. It's not as simple as we're sitting here and then we hear something and then we move our head. A lot of time it's more active the way we seek information in the environment. But in a simplified way, for the purpose of this course, let's assume that this is how we interact in an environment. Now, every relationship here, we could study that relationship. We could study the relationship between a stimulus and your perception. For example, I could have a sound here, and then you tell me, okay, is this like play two sounds and say which one's louder, or flash two lights and you say which one is brighter. So those are under, for example, psychophysics. Today, we're talking about neurophysiology. And neurophysiology is really the relationship between an environmental stimulus and the processing that happens in the brain, the neural activity in the brain. So this is the topic of the next couple of weeks, basically, that we really want to know. When you open your eyes, for example, and we get to this later on, you open your eyes and light comes through your eye. What happens to that? What happens to that, right? This process of transduction first happens, but then you're going to have neural activity. Your brain going to respond to that. And this is what we're trying to kind of look at. Yeah, this is just a high level thing for you to just contextualize what we talk about. Just think about this cycle that we are in an environment, and then the relationship between things that we're interested in. As I said, so for example, perceptual neuroscience is really the relationship between brain activity and perception, right? So what we're talking about in neurophysiology does not really have to do with perception, right? A lot of the experiments that people have done in this domain is with anesthetized animals, right? It's like basically you put a cat to sleep and then you open their eyes and then you flash a light in front of them in different places in the space and measure the neural activity. So you don't even need them to be awake. So it's not really a perception of the cat that you're measuring. You're just looking at, okay, what happens in the brain of this cat when I flash a light in front of it? So that's what we call a neurophysiological. Obviously, ideally, you want to study this in an awake, like behaving animal, but that becomes very difficult. So people have done a lot of work with anesthetized animal. So again, you're gonna get a more in-depth view of this from Ellen, but I'm just gonna say this is a macroscopic view of the brain. These terms, again, in this course, we're not going to worry about them, about how you describe different parts of the brain and whatnot. It's just good to know the basics, right? Uh, Basically, if the eyes are in the front, right, the visual input comes in, the visual input goes to the back of your brain. So that's the posterior area of the brain. Talking about vision as an example. Then the input projects forward ventrally, which is kind of down within the organism. It's in the front of the organism. And then dorsally, which is the back and above. So this is just a macroscopic view. As I said, you do not have to worry about these terms. So I'm not going to ask you what they mean or which part of the brain refers to this or refers to that. It's just have an idea of what different organizations are. We'll talk about like the different lobes of the brain, what they correspond to and everything, but these details are not important. I just wanted to put this figure to tell you that you don't have to worry about this. Yeah. Because people get scared of these terms and they're just different ways of referring to a 3D object. But today we're taking a more microscopic view, right? So any tissue, any living organism is made of cells. And the cells that make up our brain are called neurons. And they are densely packed. Now, this is an estimate, if it means anything to you, in 1 millimeter cube of the brain, there are basically 10,000 neurons, right? So people have estimated that there are 10 billion neurons in our brain, or tens of billions of neurons in our brain, 100 maybe, like those are where the estimates are. And that's a huge number. It's much smaller than the national deficit, but it's a big number for all this stuff that are in our in the skull. And each of them, right? Each of them is a very complex. Even a single cell is extremely complex, right? People would say that even a single cell is like a machine in terms of how it self-sustains itself, how it uses energy, how it gets certain input and interacts with other cells. And it truly is a magnificent, it's really magnificent how it could all function together. But the way we think about a cell in this course is an information processing unit. That the brain is an information processing system, and these are the units building up that system. These are the units that build up that system. So what is the architecture of a cell? And again, I'll come back to this. So something like this. And again, this is a scheme of it's a schematic, right? It's not like so there is enormous variability between different neurons, right? Depending on where they are. And in this macroscopic view that I'm showing you, right, you could still see the cerebellum, and then you see the cortex, and then there are a lot of little areas in between, which is our older brain, our ancestral brain, like the brain stem, supracortical. That's like, so some organism, their entire brain is like in a small area that is in human brain, right? And they do everything with that thing. And over many years, many uh, thousands of years, millions of years of evolution, we really added layers and layers on top of that old brain. And the latest layer being added is the cortex, which endows us with all our cognitive abilities, basically, with higher level cognition. Um, so that means there is enormous variability. Neurons do not look like each other necessarily when you look at a neuron in the cerebellum or you look at a neuron in the cortex. But the way we discuss them in this course are usually what's called pyramidal neurons, and they are in the cortex. And that's what looks like this. And this is the neuron most people get introduced to. Again, you have, as I said, it's a complex system, right? It's a cell. It has to self-sustain itself. It has to use energy in a certain way. All of those things. We're not going to worry about that. What we're worried about is that the cell is an information processing system. It takes certain input, it does something to them, and then it provides certain output. So think of a cell as this idealized machine where it takes input through its dendrites from other cells, and then it passes on to other cells the information through using its axon. And then the axon terminals connect to the dendrites of the other cells. So for example, in this, again, this schematic is that you're saying, if I have a receptor, and the receptor is where a physical energy interacts with a living organism. And what does that mean? Well, if I want to perceive light, I have to have a way to perceive that light. Light is just an energy. So, for example, when you take a film, like imagine an old school film that you wanted to develop, why did the picture actually appear on the film? Well, the film had a certain material that interacted with the light. A chemical reaction happened when the light, when you take a picture, again, think old school, not digital. Think old school. When the light hit the film, it created a chemical reaction there, basically, that changes the material of the film. Something like that happens in the eye. When the light hits the eye, it causes a chemical reaction, and that chemical reaction is translated into neural activity that is passed on to other neurons, and then passed on to other neurons, and passed on to other neurons. So this cascade of information travels through your brain. Okay, so the terminology, what do I have here? Okay, so the terminology that we use here is basically that the dendrites are where a neuron receives information, and we'll talk about how what's the nature of this information, and it passes on the information through its axon to other cells. Yeah.
You said receives or receives?
Receives. Yeah. We talk about what the nature of this information is. How do these cells communicate with one another? But I want you to get the general framework right. The modern psychology of the brain says that cognition is computation, that everything that you perceive is a result of computation happening, that you're computing something about the world. But that again, why is that? Well, it is always the case that psychologists, cognitive scientists use the latest technology as the metaphor for how the brain works. When it was before computers, they used many years ago, they used telephone system, the way the telephones are connected to one another. Many years ago, it was maybe a steam engine, and so on. So it's just there is some truth to all of those metaphors, right? The connectivity in the brain is resembling some connectivity that you see maybe in the telephone system or some other form of connectivity in the world. But the computer metaphor is much stronger than all of those things. Now, again, it's not that I'm saying that brain is like a computer of a laptop necessarily, but the idea that you have to compute information in order to be able to grasp this thing, to be able to recognize the face of someone as someone you already knew. All of those things happen through computation. So the way we talk about the nervous system, it's all going to be in the context of computation. How do I make that pane reappear? Oh. Okay. So this is as we were talking about, this is a cell, this is a neuron, and it has a complex physiology, as you could imagine, in order to sustain itself and in order to be able to communicate. So one thing that is important in the functioning of a neuron is the fact that it has a semi-permeable cell membrane. And basically it looks something like this, that you have these molecules, like a phospholipid molecules, and they are kind of like oily molecules, but what they do, they don't let everything come and go, but they have these gates where ions basically can move in and out of the cell. And the ions that are in and around the cell are usually these ions. There are other ones. It's a complex system, but in a simplistic way, imagine these are the three types of ions that we have around the cell. Now, again, I don't want to scare you with ions and polarity and whatnot, but imagine, like, it's basically NaCl is basically salt, right? Like, salt that we eat, but you dissolve it in water. Basically, that's what it is. And that's why we need salt, I guess. But these membranes, they have that semi-permeable property where the ions can travel back and forth. Basically, there are what we call ion pumps. But at a resting state, the cell has a ton of potassium, but it's just positive ions, and chloride, which is basically negative ion. And then you have sodium and chloride outside of a cell at a resting state. But when a cell starts getting, and again, so the way that the resting state is, I forgot to mention this, is that inside of a cell has the ratio of positive to negative inside of a cell at the resting state is much smaller than outside of a cell. What that means is that you have a lot more negative stuff inside of a cell versus positive stuff. And relative to outside of a cell, or relative to a ground level, the inside of a cell is very negative. Very negative, if like basically minus 70 millivolt of negativity, right? Basically think of it as like a little battery or something, like you have different charges on each side. This is a very small difference. For a cell is a lot. But inside of a cell, think of it that has generally a negative, at a resting state has a negative voltage. And that means there are a lot more negative ions inside of a cell versus outside of a cell. Now, one thing, when the cell gets excited, and we'll talk about how you excite the cell, it could be because it's a receptor, something fell on a receptor and it's communicating to the cell or something. But when a cell starts getting excited, sodium ions rush inside of a cell. And these sodium ions are positive. So when they come inside of a cell, they could counteract all the negative stuff inside of a cell. And that increases the voltage inside of a cell. Now, when the voltage goes, again, this is as high as it would go. But generally, if it goes even up a little bit to maybe around minus 40 millivolt, it suddenly explodes. Like, because a lot of these sodium, gates, imagine they're basically gates that let a sodium go back and forth. When a lot of them are voltage gated. So when the voltage goes even around minus 40 or something, they just all of them suddenly open up. And when they open up, it allows rushing a lot more sodium inside of a cell to the extent that that there are more positive ions inside of a cell than there are negative ions. When that happens, that opens up, again, potassium ions at that point. And the potassium ions, they're positive too. So when they leave the cell, because as we talked about, there are a lot of potassium inside of a cell. When they leave the cell, they bring the cell back down to minus 40. Now, this process is called an action potential. If you ever heard of the word action potential, is this process of sodium rushing inside of a cell, making the cells inside positive, and then potassium rushing outside of a cell. Now, at that point, you have a lot of sodium inside of a cell and a lot of potassium outside of a cell. But at the resting state, the potassium has to be inside of a cell, sodium has to be outside of a cell. So there is a refractory period then, where the cell basically pumps out of this stuff back down. The in and out here are, so basically above is within the cell, below is outside of the cell in this weird plot. But what it does is that the refractory period basically pushes the cell back to the resting state, right? And this happens by the sodium potassium pumps. So if you look at the whole process, the process of what we call an action potential, and this is core to the functioning of a cell. As I said, cells are information processing units. What they need to do is that they have to detect something, and then they have to communicate that to other cells. And the way they communicate that to the other cells is through action potential. That's the only thing a cell can do, basically. The only thing a cell can do is to either be quiet or emit an action potential. And this happens very quickly. This whole process that I described, from the very beginning of it, where the sodium rushes in and then the potassium rushes out, the whole process takes about, and the refractory period is all basically 1 millisecond, something like that. So a cell could do this up to 1,000 times every second, which is remarkable in many ways, but also it's limited in some ways, right? You cannot really communicate. If you know, the clocks of a computer are much faster than that. But the number of times that the cell could have action potential is limited in that sense. So if you look at a cell, it doesn't have only one action potential. It's not like that the activity of cell goes up and then comes down and that's it. No. The cell has to communicate this action potential to to the other cells that are downstream. Yes. In real life, yeah. So I actually have a video ready here that I wanted to show you here. So when I was talking about these action potentials, when I talk about these action potentials, you could actually, so what is this? So if you put an electrode, imagine a very thin electrode that touches the membrane of a cell, right? It's very thin, and this is what neurophysiology is done with. So if you enter that electrode somewhere in the brain, let's say in this case you enter it into the visual cortex, and put it next to a cell, you could actually record the electrical activity of that cell. You could see when the electrical activity goes up and when it comes down. And then you could actually hook that up. But how big is that? Well, you're going from negative 70 to, let's say, around 40, 30. So the change is around, say, 100 millivolts. And it's really small, but you could hook that up to an amplifier, and then you could actually then connect that to a speaker and listen to the sound of that action potential. So when a cell's activation goes up and then comes down, you could hear a little bop, for example. And a lot of the early neurophysiology was done that way. Um, we'll talk about these things later, but since you asked that question, I'll just gonna show you. You can hear the sound, right? You can hear the sound well. So every little bop that you hear there is basically an action potential. This is one cell. So keep in mind, we're talking about tens of billions of cells in the brain, right? So imagine what and cells, unless you're dead, the cells are actually have some level of activity, right? Every cell has a baseline activity. It's firing with a certain, like, maybe if its baseline is once every second, maybe its baseline is 100 times a second, and that's just baseline. That's just the cell doing nothing, basically, or we think it's not doing nothing, but it's doing a lot of, other stuff like regulating body activity and those sorts of things. But even without projecting a stimulus, even when you're sleeping, your cells are active. So a cell has a baseline activity. However, when the stimulus, when they're in a certain environment or certain input, or when you're ruminating about something, for example, or you're imagining the face of someone, then they would be more active or less active than their baseline. It's not like they're quiet and then they get something and they become more active. Here, it's a visual cell, and it's from a cat. And I showed you, as I showed you, there's a cat sitting, it's anesthetized, their eyes are open, and then the visual stimulus is in front of them, and someone is moving a flashlight. This is what you're looking at. So, and then you hear that sound that you hear is basically action potentials. And this cell, as you can see, is responding to light in that specific part of the world. And we talk about this. These are called retinotopic marks. We'll talk about them later. So that's what an action potential is. Does that answer your question? But no, what, you don't sound convinced.
Yeah, I guess like yeah, like I'm reading something about like emotion.
But so, as I was saying, let's say you have, just imagine that you have 100 billion neurons, right? And each of them is doing this, right? What does that mean? That means that we're talking about a really complex system. You talk about the entire population of Earth. Each of them is doing something, right? 70 of them are in this room, for example, out of the 8 billion people on the planet. 100 of them are at a concert, right? 200 million of them are forming a country, right? So imagine the complexity of how these neurons are interacting with one another, like the way we interact in the social media, right? Every person has a certain friends, and they get input from certain people, they form certain beliefs, and they pass on this belief to other people. So a single neuron doesn't, right? You can't decide the election by the beliefs of a single person, right? Because the single person is just a single person. It gets information from a few hundred people, and then maybe it passes on information to other people. An emotional state, let's say it's like an election, right? It comes about from collective behavior of maybe a million neurons, right? A million neurons are collectively representing, and we talk about this later, which is like a population coding. But a single cell gets input from certain number of cells, let's say around 1,000 cells, passes on this action potential to another cell. Questions?
exchange, is that just specific to neurons, or does that happen in other cells of the body?
I think,
Other
do you want to answer
cells,
that?
yeah, there are other cells that use a chemical gradient, so pass ions back and forth. But neurons are the only cells that can propagate and use, basically harness that potential to propagate an action potential all the way down the axon. So other cells do use it as part of other processes, but the only cells that use it for specifically communication are neurons.
Yeah, I imagine, so I'm not an anatomist myself, but it could be, yeah, it's an interesting property to have in terms of, yeah, I didn't think it was used that much.
Some glia, I think, are
Glia
like,
in the brain.
yeah, some
Okay.
glia in the brain are less chemically
Yeah.
active. And then
Yeah, I think those also pick up a lot of ions that are hanging out.
and then muscle cells also obviously use it, but
So speaking of muscle cells, so imagine, again, this is a schematic. Not one neuron doesn't control a muscle, right? It's a collection of neurons. But imagine if this cell was a cell that wanted to control a certain muscle. So you can imagine that if there is an action potential, this action potential has to travel the length of an axon. And some of these axons are really long, that connect different brain areas. And in order to do that, you have to have action potentials, multiple of them. So they start from what we call the axon hillock, which is basically connected to the soma. And then it goes on, and these are the myelin basically covering the axon. So it doesn't die out. So you would only need action potential at the areas that there are gaps. So you come here, and then there's another action potential, and that process continues until we reach the target. Now, the target could be a muscle, it could be another neuron, it could be, as I said, 1,000 neurons to get the input. Was there a question back there you wanted to ask, or? So, and some cells use that information. So there are different ways that cells can communicate. We're focusing on only this type of it because it's much more common. But there are like electrical stimulation and other stuff like that. But we're focusing on only this. And the way we think about it, or the way we approach it in this course, is we think of it as a way that a cell would communicate with another cell. Yeah.
What's the relationship between the myelin?
The myelin sheath basically covers the axon in order to, so you wouldn't, because it would, the action potential would kind of die out if it wasn't for the myelin. But there are gaps to allow this action potential to kind of rejuvenate and then travel down it's a complex process. Yeah, so basically the action potential starts at the axon hillock and then moves down the length of an axon, basically the wave propagating. So if you record any of these locations, you'll see that you have an action potential earlier on. Basically, when I say you have an action potential, it means that the cell activity, the cell voltage goes up and then comes back down. And then it basically travels to the next one. And then when it reaches the very end, which we call the terminals of this neuron, we call them the presynaptic terminal. So I'll explain that. So when they reach the terminal, how can they now pass on this signal to the other cell, right? This is what we're going to talk about. And this is in the context of neurotransmitters. So imagine this is the axon that you just saw. And it has traveled, the wave have traveled, and these are the myelin sheaths, and then it reaches the very end of this axon terminal, right? And this is where this neuron is connecting to what we call the postsynaptic neuron, because this is the end of the synapse. So the terminology means well, this is presynaptic because we haven't reached the synapse yet. And the cell that is on the other side would be postsynaptic, pre and post. And there is a small gap between the presynaptic and postsynaptic cell, and this is called the synapse, and the gap is called the cleft, basically. What happens here is that the presynaptic cell releases what we call basically neurotransmitters, and you've heard about neurotransmitters, and even if you don't have a science background or you heard them in the context of different, depression drugs or neurotransmitter inhibitors, like dopamine, for example, serotonin. Those are neurotransmitters, and those impact neural communication. So when the action potential reaches the axon, basically the synapse, it releases neurotransmitters. And those neurotransmitters go and bind to the ion channels of the postsynaptic cell. Yeah.
Does terminal branches just refer to the last axon on that chain?
What's that?
Does the phrase terminal branches up there?
Uh-huh.
Is that just the last axon on that chain before it's the synapse?
So like here, so when you go with an axon, it branches out. And there are many, like around 1,000 terminals at the end of the axon. Sure. So there are around 1,000 of them, imagine, at the end of the terminal. And here, I'm only showing one of them. Imagine one of them connects to one cell. So it happens that one cell may have many synapses to another cell. Like, it doesn't have to be different cells. It could be the same cell that are connected with the other cell. And that's what we call the connection strength. The number of axons, the number of synapses that are between two cells. Okay, so if a cell has, let's say, 1,000 synapses, it could be that tens of them or hundreds of them is just another cell. It doesn't have to be necessarily different cells, postsynaptic cells. Okay, so now what neurotransmitters do when one of them is released for the postsynaptic cell, it takes this input, right, as at its dendrites. So the terminology is a cell gets input, a neuron gets input in its dendrites, and then it passes on through the axon and then synapses into other cells. What the neurotransmitters do, they basically bind to the ion channels of the other cells. Why would that be the case? What good does it do if the neurotransmitters bind to the ion channels? So what purpose does it serve? Those ion channels are gating the flow of the ions, right? So if I say the only way that a cell can communicate is through action potential, and I'm saying that in order for a cell, for the activity to actually do something, it has to propagate in the brain. What is it that one cell wants to do to another cell? Penetrate. So what would it, yeah. Sorry, go ahead.
Does it change the flow of potassium and
Yeah, so if you want to impact the activity of another cell, you would want to change the flow of so let's say this cell wants to excite the other cell, and exciting another cell means that you want to cause it to have action potential. What should this cell do? What should be the impact of this cell on a neurophysiological level? What kind of impact would cause the other cell to be active?
sodium?
Yeah, so if whatever neurotransmitter this presynaptic neuron releases, if that neurotransmitter binds to some ion channels and opens up the sodium gates, what would happen then? Well, the sodium will rush into the cell, the postsynaptic cell, and increases its activity. Now, if enough cells, like it's one ion, one synaptic activation is not enough, right? You have to have many neurons activating a postsynaptic cell in order for that cell to open up enough sodium channels for its activity to go up and up to have an action potential. Does that make sense to everyone? So if I wanted now, cells could have two different types of effect on each other. They could excite one another, but they could also inhibit one another. How would I inhibit? So what kind of, let's say the neurotransmitters are released here. What are they supposed to do in order to inhibit the postsynaptic cell? Yeah? Don't bind?
They wouldn't bind, they need the uptake.
Yeah, but if they don't bind, they wouldn't change the baseline activity, right?
But you're talking
Yeah,
about inhibiting.
Yeah, yeah.
Blocking the ion channel.
So let's look at this figure again. If I want to bring down the activity, the voltage within the cell, right? The way to inhibit a cell is that you want to even bring the cell's voltage below 70, minus 70. Well, the way to do that is that you open up the chloride channels. If I open those up, then chloride rushes inside of a cell. And then the activity, the voltage within the cell would be even more polarized. So this is a terminology that I should have introduced you to. So this process of going from minus 70 up, this is called depolarization. Because initially it was polarized. The cell at the resting state is polarized. The activity within the cell doesn't match, the voltage within the cell doesn't match the voltage outside of a cell. So the cell is polarized. If this voltage is becoming closer and closer to the outside of a cell, it is depolarizing. If it was going down, it would be hyperpolarizing. So it's already polarized. Let's go even further, and that would be hyperpolarization. So cells could, yeah.
Is hyperpolarization only if it's going less than 70 or going lower from any point that it's at?
Hyperpolarization is you have to go even lower than minus 7. Okay. That the difference between the inside and outside is bigger than 7. So cells communicate with one another. Now think of, so far, the way to think about it, and I just want you to have an intuition of what does it mean to communicate. A cell can only have an action potential or not, right? There's nothing else a cell can do. It's a very simple system. Now imagine a cell is like someone very active on social media. This is my metaphor. So and let's say their baseline activity is maybe 2 posts a day, right? You have a friend who on a normal day posts 2 things. Well, they could become more active, but they could also become less active, right? So as I said, and if they don't post anything, you would think they're dead, right? So imagine, use that metaphor. So they have a baseline activity. A cell has a baseline activity. For no reason, they fire, let's say, twice every second, for no reason. But they could go above that. They could post, your friend could post 10 posts a day before an election, or they could just not do, they could go down from that. They could post maybe 1 post a day or 1 post a week. So the baseline activity of a cell is similar to that. There is a baseline activity which is normal, but their activity could go up, their activity could go down. And we call that firing rate. the firing rate of the cell could go up from a baseline. It could go down from a baseline. Well, how would it go up from a baseline? Well, if it gets a lot of excitatory input from other cells, it means that if there are a lot of presynaptic cells to this cell, and what does that mean? There are a lot of presynaptic cells. Well, there are a lot of cells that synapse into the dendrites of this cell. If there are a lot of excitatory presynaptic cells and that are active, they're going to cause the sodium channels of this cell to open up, and they're going to make this cell active. So this cell would go 10 spikes a second instead of 2 spikes a second. That was its baseline. It could go to 100 spikes a second. On the other hand, there are cells that are inhibitory. That means that the neurotransmitters that those presynaptic cells release reduces the activity of this cell because they open up channels that rushes negative ions into the cell. And as a result, they don't allow this cell's activity to go above a certain level in order to actually reach a certain level to have an action potential. So imagine this is the cell you're interested in. You could have excitatory input, you could have inhibitory input, and this excitatory and inhibitory are a collection of, let's say, 1,000 cells. These are not just from one cell. They could inhibit this cell or you could excite this cell. If inhibition and excitation are the same, this is what we call baseline activity. And at baseline, the cell has some firing rates. If the excitation goes up and the inhibition go down, the cell's firing rate goes up. And each of these lines are an action potential. Each of these lines is one of those bop that you heard in the video. Right? That's what an action potential is. If the inhibition goes up and the excitation goes down, the cell goes quiet. Right? You could inhibit the cell to a level that doesn't emit no action potential, or you could really hyperpolarize a cell for a short period of time. And a cell now compute. So now the idea of computation again comes back to this. What is a cell computing in this scenario? Well, the cell is computing, it looks like it's computing a sum of its excitatory and inhibitory inputs and averaging that and then communicating that how does the cell communicate? By its own firing rate. So the cell is saying, well, I'm getting some inputs that are exciting me, but those are more than the stuff that are inhibiting me. So I can compute something about the environment and then pass it on to the other cells, right? And again, imagine this is someone in a social media. They have their connections. There's somebody that always, their posts, brings you up and makes you be more active on social media. And then occasionally you come across something that brings you down and you're like, I don't want to be on this website or something like that. Imagine like that. And your activity is a summation of all those inputs that you're getting. You're getting excitatory input, you're getting inhibitory input, and then that defines your activity. Questions? Is this, if you don't have questions, it means that you're all understanding what a cell does.
Do you mind repeating? You were saying cell firing rate goes up when
So let's say in a car, right? You have a gas pedal, you have a brake pedal. Okay. If I push on the gas pedal, what happens? The car goes fast, right? The firing rate of a cell goes up. If you pick your foot up from the gas pedal, the car goes slower, right? Now, there is another way to make a car go faster other than pressing the gas. So let's say the car is going at a certain speed and I'm just pressing on the brake pedal. If I pick my foot up off the brake pedal, what happens? Well, the car goes faster, right? What am I doing? I'm lowering the inhibition on the car. So there are four different ways that two cells could communicate, right? So here, the reason, for example, and again, I'm showing some excitatory input here, but part of the reason why from E to D, the activation goes up is because the inhibition is getting smaller. It means that you had your foot on the brake pedal for a while. Now you pick your foot up. You're lessening your inhibition of the car. So the car goes faster. Does that make sense to everyone? Alternatively, you could have kept your foot on the brake pedal and then just gassed it up, right? Then you could also increase your car speed or your firing rate, you know? So there are two different pedals, imagine. And one of them is inhibiting and one of them is exciting. And you could either have a lot of inhibition or a little inhibition, or you could have a lot of excitation or a little excitation. And all of those things can impact the activity of the postsynaptic cell. Yeah. So the way the two cells communicate, as we said, is through these neurotransmitters. What defines a cell's connection to another cell as inhibitory or excitatory, whether it's a gas pedal or a brake pedal, is the type of neurotransmitter that it releases. So if a cell always releases excitatory neurotransmitters, it's always going to excite the postsynaptic cell. If a cell always releases inhibitory neurotransmitters like GABA, then it inhibits the postsynaptic cell. So what is the physiology of the cell defines if it's an excitatory presynaptic cell or if it's an inhibitory presynaptic cell. Yeah.
Is the physiology of the cells, is that a biological or is that, are there ways to change that?
That's what a cell is, like basically.
Right.
Yeah, the cell is either excitatory or inhibitory and it doesn't change. If a cell is excitatory, it's always releasing like glutamate, like an excitatory basically neurotransmitter that opens up positive ion channels in the postsynaptic cell and allows for the activity of the cell to go up. And if it's an inhibitory cell, it always releases, the type of neurotransmitters that inhibit the post cell. But again, it's important. People forget the cell is always have some baseline activity. It's not dead. So if it has a baseline activity, you could still inhibit it. You could still bring it below the baseline activity, or you could take it above the baseline activity. Downstream cells or postsynaptic cells, and again, imagine you go for example, for activity from the eye to reach your face selection area, you go through 6-7 layers of cells. And this is how you pass it on. You pass on the light to a certain area that we'll talk about, to other area, another area, another area. And you just go on within 6, 7, 8 communication, you reach, for example, face selective areas. And some of them are exciting the other ones, some of them are inhibiting. And this happens on a synaptic level. And you have, let's say, if you have 100 billion neurons, you have 100 trillion synapses, right? There's a lot going on here for the computation to take place here.
When you like drink coffee, you're not changing the cell, you're just changing like the process.
Yeah, you're not changing the cell, you're changing the uptake of these neurotransmitters. So you're kind of changing the excitement. So if you have a lot of, let's say, excitatory neurotransmitters in your brain, it makes it a lot easier for one cell to excite another cell, right? Because you already are predisposed to this excitatory influence. So when you take a drug like a serotonin reuptake inhibitor, right, what happens is that it allows serotonin to hang around, and that creates this easy communication between the cells. opens up different pathways that were initially inhibited, for example. We'll talk a lot more about computation in this course because I like it. And we look at how you could, for example, have these cells connected to each other with inhibitory and excitatory stuff, that it computes different stuff, right? It computes, for example, it starts from very simple computation. Oh, that there is a circle of light here. If there's a circle of light and you want to detect that, well, if you have some excitatory cells in the middle and some inhibitory surround, then you could detect the light. So with this is the building blocks of the brain. These just excitatory and inhibitory connections between the cells. And with a lot of them, you could create a general purpose computer like our brain. The fact that cells can communicate and they can excite and inhibit each other, they could go from sensory input of a pattern of light to a perception. And we'll talk about that entire pipeline. But it's good to know the basics of how cells communicate. All right, 7:50. Great. See you all next week.
If you didn't initial the sign-in sheet, come tell us that you're here.